please read the question carefully. It is, which of the following is false? Okay, let's see the answer. So it's track horizontal gene transfer. I know there is some confusion with option C, that estimate microbial diversity. So here the phylogenetic tree does estimate microbial diversity. Okay, so this is true statement. We are not talking about quantification. Okay, phylogenetic tree cannot quantify microbes, but it can though estimate microbial diversity. So when we make this phylogenetic tree, we can see from how a common ancestor, the given number of the given bacteria or the given diversity of bacteria emerges. Okay, we cannot quantify, but we can always estimate microbial diversity. So that thing is very important. Just read the option very carefully if it is talking about the qualitative assessment or the quantitative assessment. So here, if I write quantify microbes or quantify microbial diversity, then it would have been a false statement. Okay, and yes, it cannot track the horizontal gene transfer. So that thing is there. Moving towards next question. So please try to solve this question. You have to just match the following. NIFH 16S rRNA gene, and there is one conversion of ammonium to organic ammonia, and the second is organic ammonia to ammonium ion. Okay, this ammonium NH4 plus is in dissolved form. Okay, please keep that in mind, just an extra information. Anything which is in ionic form, okay, anything which is in ionic form, okay, ionic form means there is sign plus or minus, that is in dissolved form always. When there are no charges present, then we can say it is either in gaseous form or just a elemental form. But as you can see, there are charges in ammonium NH4 plus. We never write NH4. Okay, it means the option number 4 that is shown here, it's wrong. It should always be NH4 plus. So that is the correct way of representing. So take some time to solve these questions. Okay, I can see pretty much responses, so we'll see. So the correct option is, as you have suggested, it's option A. Yeah. So that is the correct one. So the thing is, when we take out something from the dissolved form and make structure out of it, when we say organic nitrogen, it means it is in form of some structure, like the green thing in the leaf, that is nitrogen. So if we take a leaf, it is just fallen from a tree or it is intact in a tree, it contains more of nitrogen and less of carbon. So when we talk about green side of the leaf, it is nitrogen and mostly in the form of organic nitrogen. When with time this leaf dries out, it loses its nitrogen and we can see more of carbon. So the brown thing is the carbon. We cannot burn green leaf. It's difficult. One, because of moisture, and secondly, because of this nitrogen. However, it's easier to burn brown leaf because it contains carbon and it is a good fuel. So we can get more energy out of when we burn this brown leaf. So it is this kind of nitrogen is in organic form. So that is why it's organic. So taking ammonium ion from the water and preparing a structure is called assimilation. And this conversion of organic ammonia into the dissolved form or the ammonium ion is called ammonification. And other things are very simple, you know. Moving towards next question. This is the graph that I have given, a procedure I have given for you to understand that what is ammonification? Conversion of organic ammonia to ammonium ion. You can see very clearly here NH4 plus is there. And here also you can see NO2 minus and NO3 minus is there. So all these are in dissolved form. Okay, so this thing is very important to understand that ammonia, this sign indicate that they are in dissolved form. So this, due to ammonification, we can convert organic ammonia, which can this is organic ammonia, so this is in the solid form. And when we write just ammonia, okay, it is in gaseous form. Ammonia is a gas, a pungent smelling gas. However, organic ammonia is in the solid form. So here organic ammonia, which is in the solid form, it undergoes the process of ammonification and it gets converted to ammonium ion, which further there will be in the presence of aerobes, it undergoes aerobic nitrification and it's converted to nitrate via conversion to nitrite. So this nitrite is very transitional form, so it's not very stable, so it does not exist for quite long time. Then it is converted to nitrate. So I can give you one question here. You can write what type of bacteria converts ammonia to nitrite and what type of bacteria converts a nitrite to nitrate. You can answer this in the chat box. This is a very simple question. A group of bacteria are there, right, which carry out the nitrification process. So what are the name of those groups of bacteria? Am I not audible? Yes. I am audible, right? Yeah, this is nitrifying bacteria that is there. Okay, I'll repeat all the questions again. Do you want to know the second question? Okay, I'll go back again. Let's first solve this question. My question is that what are the categories of bacteria that converts ammonium to nitrite, then to nitrite to nitrate. So this whole category comes under the nitrifying bacteria, but there are two subsections for those particular task. So what are those called? Yes, Perry Croslin has answered. It's Nitrobacter and Nitrosomnas. So now Nitrobacter is for which process? Nitrite to nitrate or for ammonium to nitrite? Please answer this. This is for what procedure? Ammonium to nitrite or nitrite to nitrate? Your answer is correct. Nitrobacter and nitrosomonas only. But could you please mention their specific task? You can take some time and mention. See, even if you are using internet, if anyone is using internet for solving these questions, there is no problem. It's not a test. But I just want to inculcate this knowledge of this thing. So if you, even if you take time to check all the answers in the internet, that is good. You will learn more. Yes, ammonia to nitrite is nitrococcus. Yes. Okay. So I would like to answer here from the group of bacteria which are employed for the conversion of ammonia to nitrite are called nitrosomonas. Okay. So nitrosomonas, they convert ammonia to nitrite. And the second category of bacteria which are used for the conversion of nitrite to nitrate are called nitrobacter. So the sequence that you have written is just opposite. Like you should first write nitrosomonas and then nitrobacter. And nitrococcus is also correct. Okay. So nitrobacter, nitrospina, nitrococcus, all these are used for the conversion of nitrite to nitrate. Okay. Nitrite is NO2 minus and nitrate is NO3 minus. Okay. So there is one more procedure which was later found, that which converts directly ammonium ion to N2 gas. So N2 is harmless gas. So our major aim is the conversion to N2 gas. So this process is called anamox, anaerobic ammonium oxidation, anamox. And what is nitrogen fixation? It is the conversion of, like, it captures nitrogen fixation is the process in which we capture the atmospheric nitrogen, or if there is any particular source of nitrogen, and convert it into organic ammonia or organic nitrogen. So that process is called nitrogen fixation. So there are many plants or bacteria that can do that. So you might have heard about the leguminous plants. So it contains those bacteria which are very good in this nitrogen fixation. So moving towards next question. Okay, I have mentioned here also, for this process NiFF, FH is there. Which of the following is most likely to be found in hot spring? Please answer this question. Which of the following is most likely to be found in hot spring? So is there anyone who can tell me why hot spring is hot? What compounds are present which make these hot springs? Yes, correct. Very true. Yes, 100% correct. Sulfur. Because of the presence of sulfur, this water is hot. It's not, because many people think that it's coming from the like it's coming from depth, that's why it is hot, but no, it contains sulfur. So in that line, what is the solution for this question? Does it contain purple sulfur bacteria, purple non-sulfur bacteria, green sulfur bacteria, or green non-sulfur bacteria. So this and green, purple, these color are the pigments, color of the pigments. Please take some time to answer this question. We'll see solution for this question. So it is green sulfur bacteria. So this purple category is always uh, absent, and this green sulfur bacteria is most likely. Okay, so read the question very properly. Here it is asking which of the following is almost there, always. So green sulfur bacteria is there. So the next question is essential element for the growth of hydrogen metabolizing bacteria in culture is, which of the following can help in hydrogen metabolization? So most of them are chelating agents. Okay, trace this and most important thing is these should be present in only trace amount. If they exceed that trace or tolerance category, then they can be poisonous, not only for bacteria but for any human body. We say, right, copper. Okay, I would like to share one very important thing. So you might have heard in your houses, or you might be practicing this also, that we keep water in the copper vessel, right? Okay, all of you have done this, right? We keep water in the copper vessel at night and we drink it in the morning. So many people are doing these things. You have done it, right? Or you might have seen people doing it. Yes, correct. So this practice, so why we do this? We think that this copper, first reason is copper has disinfecting properties. So the number of bacterial counts that were present before keeping water in this copper vessel, and the next morning after 8 to 9 hours of disinfection, the number of bacterial count significantly reduces. So this practice was initialized, it was developed by our ancestors when we were using surface water or the groundwater or water after basic treatment. Okay, but now many of our household are using RO at our homes, right? So when we use RO, okay, how many of you are using RO at your homes as a point of treatment for water? I know there are places in which this facility is not available, but in many cities, many small towns also, if there is nothing, we will keep an RO, AquaGuard, some unit at our houses, and we'll drink the filtered water from there. So I hope many of us are using this RO at our homes. So people are following the similar practice, that people are keeping this RO water in the copper vessel and drinking it next day morning. But now there are, in the past practice and in this RO water, there is some major difference. So what is that difference? Initially, when we were putting this surface or groundwater in the flask, in the copper flask, the water was saturated with minerals. Okay, there was calcium, magnesium, and many other minerals. So the water was already saturated. So the copper was coming out in very trace amount. Okay. So this copper was coming out in trace amount and it was just used for disinfecting the water. But now with this RO technology, the water is devoid of minerals. So there is no mineral. Water is undersaturated. There is no calcium, magnesium, and other minerals. The water is completely devoid of minerals. So when we put that type of water, it's like a distilled water. So when we will put that type of water in this copper flask, so it will take out the copper from the side walls and the copper will be dissolved in such water. So initially when the groundwater was used in copper vessels, only little amount of copper was coming out, trace level. But now it is coming out in higher amounts. So as I have told you already, this copper is very good for the body. It's really good, but in very small amount. When it is in trace level, it is very helpful for the metabolization. But when the concentration goes above a threshold, a certain limit, it will just accumulate inside the body. So there are many cases of the copper accumulation inside the body. So even this copper is not very good in higher amounts inside our body. So that's why never keep RO water in the copper vessel. Okay, so that thing is really important. What type of water should be kept in RO vessel? Take the government supply water, whatever you are getting in your homes, or surface or groundwater. If you are using surface or groundwater, filter it, boil it for 20-25 minutes. Then you can keep that water in the copper vessel and you can drink it the next morning. But never ever keep this RO water in the copper vessel and have it. So that is take home message from this question. So we'll see the solution. So that's why we say that RO water is not really good. It is just the marketing strategy. So the answer is nickel. Moving towards next question. Which amongst the following are the desirable qualities of genetic marker for building phylogenetic tree? Conserved and variable domains, universally present, or it contained evolution or more GC content. So which of the following are the desirable qualities of genetic marker? Please answer this question. Take some time and answer this question. We'll see the solution now. The solution is option number C, that is ABC. So the desirable qualities of genetic markers for building phylogenetic tree are conserved and variable domains. Okay, it should be universally present, and it contain evolution. Moving towards next question. Which of the we have to just match the followings. We are talking about fermentation, Calvin cycle, and respiration. So the procedure that are explained in these are oxidative pathway, autotrophic pathway, oxidative phosphorylation, and substrate level phosphorylation. We have done a similar kind of question earlier, so it won't be difficult for you to solve this question. Please take some time in solving this question. This is very simple question. Please try this. Mr. Raja Praveen, do you want to ask something? Your hand is raised. Or was that a mistake? So the solution for this question is autotrophic pathway is a part of Kelvin cycle. Oxidative phosphorylation is respiration, and the substrate level phosphorylation is fermentation. We have seen this question earlier also. Okay, there was someone who was asking about question number 2, so I'll go there first. We will see that. Then we'll move forward with question number 10. So the question number 2 is simple. We are talking about just the statement which is true or false. You can click the picture for this question. Also, I'll share all these slides with you as well as the recorded lecture. Okay. You just note down this question or take a picture. Then I'll give you a minute to do so. Is that okay? I guess. So then we'll start with question number 10. I hope it's done. So we'll move forward to the next question. That is question number 10. Which of the following are true attributes of Crocolus group of cyanobacteria? This is kind of memory based question. You need to jog your memory to solve such. It's multiple choice question. Please take some time and solve this one. We are talking about cyanobacteria, one of the most primitive organisms. Okay, so they have prokaryotic properties. Yes, many of you have answered. So that is unicellular binary fission. So this is the kind of the groups that I have written here for you to understand. So these are the 5 morphological groups of cyanobacteria. Please note them down. So Crococolus is unicellular with binary fission. Plurocapsules, capsulas, it's unicellular with multiple fission. Oscillatoris filamentous non-heterosyntheous bacteria Okay, and then other names. The names are difficult to pronounce. Non-Stokocles filamentous divide along a single axis, and stigmometals filamentous divide along multiple planes forming branching filaments. Please note this down. So this is one important concept that I wanted to explain, which is called cell lysis. Okay, this was explained earlier, but I would like to explain it again by using some pictures so that it becomes easier for you to understand. So let us imagine this is a intact cell, bacterial cell we can say, and it contains various internal organic matter. These triangle you can see are the representative of internal organic matter. Okay, so this is an intact cell. So what happened when it is attacked by any oxidant or any environmental factor? It can be anything. uh, Oxidant addition means if we are just putting anything that anything that can lead to cell breakage, anything that is used for killing the bacteria. Okay, so that procedure, so this represent the environmental factor or oxidants. So what happens with time? Due to this attack, either naturally or artificially, the cell membrane breaks down. So these dotted lines represents cell membrane breakage, and the internal organic matter comes out. So the next process is the coming out of internal organic matter. When this comes out, it ejects out, it is called external organic matter. So this is the process of cell lysis. Just wanted to give a visual representation of cell lysis. Do you want to ask any question in this of question of this hot spring? You can type the question in the chat box, and I'll help you with that in the end. So this was explained in the first lecture also, introduction, but I would like to explain that again. I can see many new faces also. In this week it was explained in much deeper details. So gram staining, it was developed by Danish physician Hans Christian Gram, so that's why we call it gram staining. So here the bacteria are categorized into gram positive and gram negative bacteria. Okay, so this is the procedure for gram staining. Here 4 type of chemicals are there, so we get all these things in a gram staining kit, so that makes it easier for us to check if the bacteria is gram positive or gram negative. So what happens, we first put crystal violet dye on the bacteria, which gives purple color to the bacteria. Then what happens, this happens in a glass slide. A rectangular glass slide is there. I hope you have seen this in school as well. So there is a glass slide, transparent glass slide. Here we first put some bacteria, then we apply crystal violet, then we heat it up a bit, like heating, like just for 5 seconds of heating, okay, over a lamp or a candle you can use. Then we apply mordant, which is in the form of iodine. We apply mordant. The third step is we wash it with alcohol. After application of mordant also we heat it for just 2 to 5 seconds. After that we use alcohol to wash this. So some bacteria will retain this purple color, but other bacteria will be washed out. It will become transparent again, or whatever the color that bacteria is holding initially, so that will take that color. So purple color will be washed out from certain bacteria. So this is called alcohol wash or decolorization. Next, when we apply safranin or the counter stain, the bacteria which have lost their color, they will retain this new color. So in the slides we can see two different colors. One is purple and other is safranin. So these things are there. So this safranin is called counter stain. So first is stain, the next is counter stain. So we can see these bacteria under the microscope, and we can easily figure out which one is gram positive and which one is gram negative. Okay, so this slide explains it better. So bacteria which does not give out color on alcohol wash, means which retain the purple color, are gram positive bacteria. Okay, and the bacteria which are washed out using this alcohol and they do not retain the purple color, but they takes up the color of the safranin or the counter stain, are called gram negative bacteria. So in this figure you can see, in the first process, when this fixation is there, fixation means just the heating, just 2 to 5 seconds of heating, so both of them are transparent. Secondly, we applied crystal violet, so that is first stain or purple color, so both the bacteria will get this blue color. Then next thing is iodine treatment, so this is also used for fixing the color. Then both of them will get this purple color. The next step is decolorization by using alcohol. So what will happen? Some of them will retain purple color and the other bacteria will be decolorized. When we use the next stain, the counter stain or safranin, the bacteria which were already purple, nothing will happen to them, they will remain as such. And the other category of bacteria which were white, they will get this color of safranin. Okay, so that's why we see two different colors. So one is gram positive and the other is gram negative. So this is the principle of gram staining. So when the bacteria stained with primary stain, crystal violet, primary stain, and counter stain, two stains are there. So the first is primary stain, crystal violet, and fixed by mordant. Mordant is what? Iodine. Some of the bacteria are able to retain the primary stain and some are decolorized by alcohol. So we are talking about step number 1, 2, 3 in the first point. Now why this is happening? The cell wall of gram positive bacteria have a thick layer of protein sugar complexes called peptidoglycan, and here the lipid content is low. So decolorizing the cell causes this thick cell wall to dehydrate. The cell wall is thick and decolorizing will dehydrate it. So what will happen? Since the wall is thick and no lipid is there, when it decolorizes, it will shrunk, it will lose out all the water content. So because of that, the pores will become smaller. So earlier if the pore size was some R, then after washing out it will become maybe R by 10, R by 100. Okay, so it will become smaller. So the pores are, in some cases, pores are completely closed. So whatever the color which was there inside, it remains it's inside. And one here very important thing is, in gram staining, bacteria are killed. Okay, the bacteria are no more alive. So this is one method of identification. The other methods are there in which bacteria remain the viability. Okay, they are alive, but in gram staining procedure, bacteria are killed. So all these procedure kills the bacteria. So that thing is important to know. So number of bacteria won't change with time in this case. So this, since the wall is thick, it will retain the color because color is not coming out when the pores are closed. Okay, so the ethanol cannot remove the crystal violet iodine complex that is bound to the thick layer of peptidoglycan of gram-positive bacteria and appears blue or purple in color. In case of non-gram-negative bacteria, cell wall also takes up crystal violet iodine complexes, but due to thin layer of peptidoglycan and thick outer layer which is formed of lipids, so in gram-positive bacteria, peptidoglycan layer is thick but lipid layer is thin, and in gram negative bacteria it's just opposite. Peptidoglycan layer is is thin but lipid layer is thick. So here what happens, this crystal violet iodine complex gets washed off. So when they are exposed to alcohol, that is decolorizer that we are using here, it dissolves the lipid layer. So in gram positive, lipid layer is really thin, so it does not matter if it dissolves or not. It's already thin, it does not matter. But in the second category, when the layer is thick, when it gets dissolved, one complete layer is removed. So anyway, in all these procedure, bacteria are killed, but here in this procedure, it is completely destroyed. So decolorizer dissolves the lipid in the cell wall, which allows the crystal violet iodine complex to leach out of the cell. So the purple color which was given by using the primary stain, it comes out. So when we stain it with safranin again, it takes the color of the safranin and it appears red in color. Okay, so that is what is happening in the gram. So that is the way to differentiate between the gram positive and the gram negative bacteria. So coming to the question number 11, identify the false statements. I hope you understood what is gram positive and gram negative bacteria. This is a repeated information, but I just wanted to share it once again because few concepts are really very important throughout the subjects, as well as even if you are going to other courses. If you are taking any course around the microbiology, few concepts are very important, so I'll keep on repeating them for just to refresh your memory. So now the next question is, identify the false statement. Please write down the correct answer for this and also mention why that particular statement is wrong. What can be the correct statement? So first you write the false statement, then the next part will be the true statement for that wrong one, the correct statement. Take some time to write the false statement here. Please take, I'll give you some time, some 2-3 minutes. Yes. Please also write the reason for your answer. many of you have answered option C, but I can see one option B also. Could you please mention why option B as well? Just think about the what is wrong in that false statement. Take some time. You can take some time and write the correct statement. Okay, we'll see the solution now. So the correct answer is C. Alpha helix and beta strands form the tertiary structure in proteins. Okay, so this is the false statement here. So the next question is identify the wrong pair. Tryptophan, aspartate, cysteine and triazine. Identify the wrong pair here. I can see two options here, D and B. Others can also try. Okay, we'll see the solution now. So the option B is the wrong pair. Aspartate, it's showing positively charged amino acid. So what can be the correct answer for this one? What is aspartate? Yes, correct. It is negatively charged amino acid. So moving towards next question. So here also you need to identify the false statement. We are talking about tryptophan, stop codon, start codon and wobble base pairing. So please try to find out the false statement here and also give the correct one or what is wrong in that statement. Give it a try, everyone. You need to identify the false statement. I didn't get any answer till now. Okay, I got one. Others also, please give it a try. Those who are saying option A, correct this statement also. What is wrong in this one and what can be the correct statement? And for option D also, what is wrong in this one and what can be the correct statement? Give it a try. What can be the correct statement? What is wrong in those statements? Just write down in the chat box, no problem. Even if you're wrong, no problem. Okay, so yes, D option is wrong. As you have said, wobble does not cause mutation. Okay, so it does not cause mutation, so that's why statement D is wrong here. Option A is correct. Okay. So the next, okay, you can see as we were studying the structures of protein in the earlier questions. So primary structures are the sequence of chain of amino acid, nothing else, just a sequence of chain. They do not cause anything, just a sequence. The secondary protein structure, what are those? Alpha helic or pleated structure. So what are those? They are hydrogen bonding of the peptide backbone causes the amino acids to fold into a repeating pattern. So a sequence of amino acid there. So the repetition of this sequence is the secondary protein structure. So it can be pleated sheet or it can be alpha helic. Then here next comes the tertiary protein structure. It is three-dimensional folding pattern of a protein due to side chain interaction. Okay. So primary protein structure, then secondary protein structure. There are two types. One is pleated and the next is alpha helic. The third is tertiary protein structure, which is three-dimensional structure. The next is quaternary protein structure. It consisting of more than one amino acid chain. So that is quaternary protein structure. You can note this down. We'll move forward to the next one. So this concept is also very important in microbiology or the biomolecules. How these amino acid sequence, all these things are arranged. Once change in arrangement, a completely different protein will be formed. coming to the next one. In logistic, now we are talking about bacteria growth curve. Okay, so the first question is in that category. In logistic growth model, the nutrient limitation plays an important role in determining the final population. So this is very obvious. So answer can be true or false. Okay, very nice. I need more responses. Give it a try. Yes, your answer is correct. True. Obviously, if there is no nutrients, no bacteria will grow. So, and if we are putting some nutrient, there will be growth of bacteria. So, okay. So a few things are required for the bacteria to propagate. So one is nutrient, other thing is right environment, and third thing is some initial bacteria. If the environment is very clean, means if the condition is sterilized, we are putting lots of nutrient, but if the condition is sterilized, there won't be any growth. Okay. Similarly, if there is some bacteria but there is no nutrient, there won't be any growth. Third, if bacteria is also there and there is some nutrient but the environment is not good, means we are putting like 100 degrees Celsius, 2 degrees Celsius of heat, we are putting it in fire, no bacteria will grow. So environment, food, and someone to eat that food, these three things are really important for the bacteria to grow so that we can determine the final population. So if there is less nutrients, the final population will be less. What will happen? The bacteria will eat each other for fulfilling their nutritional requirements. So this is in the logistic growth model. So this was very simple, so I'll just move forward. Here you need to identify the true statement. Low turbidity signifies exponential phase in the logistic growth model. As you know that this bacteria can be measured by using turbidity, right? So it is just the correlation between the turbidity and the number of bacteria. So the first option is low turbidity signifies exponential phase in the logistic growth model. Measurement of turbidity and the plate count are direct method for microbial cell measurement. The extent of lag phase depends on the age of inoculum. When the microbial population is lesser than the carrying capacity, microbial death starts to occur. Take some time and answer this question. I need more responses. Okay, so all of you have answered it correctly. The answer is C. The extent of lag phase depends upon age of inoculum. So what is lag phase? That is the initial phase in which the bacteria is trying to adapt to the environment. So if the bacteria has already adapted in such or in similar situation, it won't take much time to adapt in the same situation. Okay, so the lag phase, which is the adaptation phase, will be really low. So the extent of lag phase, as we can see, depends upon the age of inoculum. So the next question is, decay term in the growth model is important because when we want to quantify why do we require to use the decay term? Initially it was not at all used. I hope you guys remember the equation n is equal to n naught into 2 raised to power n. Here there was no decay term. We were just considering the bacteria will keep on growing, it won't die at all. So why didn't decay term was introduced? But we know the bacteria do die, right? So the why decay term is in the growth model is important. Please mention the correct answer. Other people, please answer. Okay, we'll see the solution now. Okay, so why do we use the decay term? Because it incorporates the removal of microorganisms due to natural death. Because we know that bacteria do die naturally, right? Maybe due to lack of nutrients and other reason. So it incorporates the removal of microorganisms due to natural death. Otherwise, the model would have just predicted the bacteria keeps on growing and growing. So this is the number of bacterial cell versus time graph. So this is called growth curve. Okay, I have taken this picture from at biology exams for you, so you can see this for more information. So here there will be an initial lag phase. So here what happens, the number of bacteria will remain same. If I am putting 100 bacteria, the bacteria will remain 100. However, the weight of the bacteria will increase. Why? Because there is increase in size. Okay, so the size increases, number remains same. You can see in this picture, right? Small bacteria are there near the curve, then it is growing to the bigger bacteria, but number 1 2 3 4 5 is there, so it's only remaining 5. Second phase is log phase. Here the number of cells increases exponentially. Here the number of bacteria increases. So I would like to mention here, in the log phase the size of the bacteria is smallest. Okay, the media is most turbid and the size of bacteria is the smallest. Here it is growing, it is multiplying by binary fission or other procedure. The next phase is stationary phase. In here the number of bacteria remains same. So in the log phase, at the end of log phase 15 bacteria are there, then in the stationary phase also 15 bacteria will be there. So this is called stationary phase. The next phase is phase of decline or endogenous respiration or death phase. Here the number of bacteria started to decrease. This is because of some natural death. And the most important thing is, here the media or the system that we are using is a batch culture. Batch culture means there is no incoming food or outgoing food. We will just take a beaker, anything, we will just put some bacteria, put some nutrients and put this system in a good environment which is viable for bacterial growth. I told you, right? Food should be there, the bacteria which will eat the food should be there, and good environment for the viable environment for the bacterial growth should be there. All these three things should be combined in a single system beaker. or any flask. Oxygen should be there, okay, if the bacteria is aerobic. Now we will just keep it in good environment, like 30 degrees Celsius or 25 degrees Celsius or room temperature, whatever temperature is good for that particular bacteria to grow. Provide a little shaking so that all the bacteria can get the food. If this environment is given, the bacteria will exactly follow this curve. Okay? And at the end, it will die because of lack of nutrients, because bacteria will keep on consuming the nutrients, so the nutrients will reduce, so eventually they will eat up each other and the number of bacterial cell will decrease. So the next thing, okay, I'll give you my website. You can contact me here. This is my website. You can just click on this, you can type it in Google, you'll get to my website. Then there you can write your question, then I will reply you via email. So when you will write the question, the website will ask to put your email. So you put your email, write your question, I will get notification and I will reply. If possible, then I will solve at that time only. Otherwise, in the next session, I can discuss that question if I need face-to-face interaction for explaining better. Otherwise, if it's simple thing, I will just solve. Okay. Anyone who wants to contact me in between sessions, then they can use this procedure. Okay. So I hope you understood this bacterial growth curve. So here, see, we have used the phase of decline or endogenous respiration or the death phase. So this is a numerical question. I'm gonna. Okay. See, when you will write the question and I will reply, then after that you can upload. You can just simply write, hi, I want to ask you a question. I need to uploads a picture. When I will reply, then after that you can upload. Before that it will be difficult to upload. I don't think you can upload it there in the website, but this will take us to the email conversation. There you can upload, but you need to check your email. When I will reply, you will get reply in your email, not in the website. Okay, so in email we can definitely then connect. Okay, thank you so much. So this is the question, a numerical question based on the growth. So if one starts with 10,000 cells in a culture with a generation time of 2 hours, how many cells will be in the culture after 48 hours? So here we know the initial means n naught. Further, we know the generation time. So the question is, after 48 hours, how many cells will be there? So here we decline phase, we won't take the decline phase. We will just talk about the log phase. So in such kind of question, we ignore this lag phase, stationary phase and decline phase. We just talk about the log phase. So please take some time to solve this question. solve this question, you can write the answer in the box when you are done. Then I will give you the step by step solution. Take some time, no problem. You can take more time if you want. You can solve this question. I can give you to 5 to 8 minutes. If you need more time, let me know. I'll give you more. Try to solve this question. Take a paper pen, also the calculator. Just apply the formula and solve this one. Recheck your solution. The formula is n is equal to n naught into 2 raised to power n. Other people can also try. Okay, since I have got many responses, I'll show the answer. Yes, the first thing is we need to know the correct formula. So to determine the number of cells in the culture after 48 hours, we can use the exponential growth equation. When we solve such questions now, mostly we talk about this log phase, unless mentioned that we need to incorporate other phases. So we just talk about the log phase. So n is equal to n naught into 2 raised to power n. Here why 2 is there? Why 2 is there? Is there anyone who can answer? And is there any chance that other than 2 we can use any other number? Yes, correct. We are just talking about binary fission. Okay, so that's why we are saying 2, because one cell is converted to 2, 2 to 4, like that. So just because we are talking about binary fission, we are using 2. If it is multiple fission, then we can use other numbers as well. So and notice final number of cells, and notice initial number of cells, which can be 10,000 or 10 raised to power 4. So 10 is to the way of writing, either we write 10,000 or 10 raised to power 4. Both are same mathematically, but 10 raised to power is more scientific. So whenever you want to write such answers, you use the scientific term. N is number of generations. Generation time is 2 hours. Okay, and total time is 48 hours. 48 hours. So the first step is calculate the number of generation. So which can be done by total time divided by generation time. We just wanted to know how many generation can be observed, can be seen in the time lap of 48 hours. Okay, so bacteria multiply really fast. So in every 2 hours they are doubling. One bacteria is is 4 in 2 hours, then 8 in 4 hours, like that. So this is the number of generations. Note this down. We'll move forward to the next slide. I hope you have noted. So the step 2 is calculate the final number of cell, just putting in this. Okay, 2 raised to power 24 is this value. When we are writing it like this, then we can write n is equal to 1.68 into 10 raised to power 11. So that is the final answer. So here I want to mention one very important thing. As I was saying earlier, one should write always in the scientific terms, because this is a science question, so it's better to write in the scientific terms. So this is called, when we write something to the raised to power something, x raised to power y, this is called scientific notation. So we should always follow scientific notation. So that is one thing I want to share. Secondly, and most overlooked thing is, see, here in this question we have written 10,000, not 10,005, 10,006, 10,100 something, right? No, I have not written it like this. It's just 10,000 with 4 zeros. So very clean number. So it does not mean the number of bacteria are this only. When we check on plate or anything, any method of bacterial number calculation, so it's not like that it will be always in this form. 10,000, very clean value. It can be 10,500 something something something. So we mostly represent by rounding it to a certain number which is in form of tens, hundreds or thousand, like that. So here 10,000 is there. So while writing the answer, like you have written, you people have given the correct answer only. Many people have given the correct answer. So like 16,007. See, this big number you don't have to write. If you are not writing in the scientific term, you can just write 17 00000 like that, or 16 00000 like that. Okay, you no need to write the big number 777216000. No. So this is called the effect of significant disease. So that looks more scientific. So when you will write 16 00000, the number of zeros that are there. Okay, yeah, the number of zeros that is there, 11. So if you will write like 168 and 9 number of zeros, then you will be prompted to write it in the scientific notation. Okay, here also see, what answer we are getting is 16, 777, 216. After multiplying it to 10 raised to power 4, see how the answer has changed to 1.68 into 10 raised to power 11. So that is the best way of representing the number of bacteria, because that can be very large. And some number of bacteria, 5,000, 6,000 in front of 1 crore is nothing, right? So we can just round up the values and represent it in the form of scientific notation. So even if your answer is correct, if you use very big number, like many number of significant significant digits, it won't look good. So try to follow the scientific notation. That will make your answer more formal, more intellectual. Okay, you take some time to note down all the values. Then I'll move forward to the next one. I hope you understood what I meant by saying it should look good. It should look more scientific and intellectual. Just the way of representing things makes you different from the people who just know numbers. Okay? When you will represent things properly, it will like showcase your knowledge. So that is all what I wanted you people to understand by answering this question. So there is one more question. Okay, you note it down, then we'll move forward. Okay, we'll move forward to the next one. So the next question is identify the correct statement. Here, 4 option of Gibbs free energy is given. You need to identify the correct one. Gibbs free energy is equal to standard Gibbs free energy and some constant value plus minus some constant value, which is the function of reaction quotient, and R and T. RT is R is universal gas constant and T is temperature in Kelvin. And be very mindful if we are using log or ln. Okay, many people have made this mistakes in the past exams, that they confuse between ln and log. Okay, all of you have answered correctly. So the answer is option B. So that's all for today. I'll keep my screen. So that's all. So if you want to leave, you can leave. But still I will keep my screen open for 5-6 minutes. In case of doubt, you can ask in the chat box. And those who are done, they can leave the chat. Okay. Thank you everyone. Thank you for being such a nice audience and such an interactive session. So thank you everyone. So always you can contact me in my website. Thank you.